Today I am going to explain about topic of viruses. So virus, the term virus originated derived from Latin language, Latin word. So meaning of word virus means poison. So viruses are crystalline inert particle in environment. These are inactive outside the host body of other host organisms. So these viruses are obligate intracellular parasites. So these are submicroscopic, ultra microscopic obligate intracellular parasite, submicroscopic. Generally microscopic means too smaller condition, so these are smaller than the bacterial cells. So ultra microscopic, submicroscopic obligate parasites. So viruses are submicroscopic, then obligate intracellular parasite, obligate intracellular parasite. That means outside the host organism, these are inactive condition, these cannot replicate. So these can replicate inside the host organism only. So for the replication of virus, host is a mandatory, that means a host is a compulsory. So without host these cannot replicate. Why? Because in viruses there is no metabolism. Viruses lack the metabolism and enzymes are absent. So what enzymes are required for replication of viruses? So metabolism completely absent in a virus. So virus is a nothing but of inert crystallized particle outside the host organism. These viruses can active inside the host only. That's why these are obligate intracellular parasites inside the host organism. So cellular structure also absent in viruses. So these viruses are a cellular condition. So without a complete cell structure, organism cannot independent existence. So viruses have no the cellular structure. That's why viruses not have the ability to independent existence. That's why these are required host for complete the life cycle. That means the production of virion particles more infectious virus particles. So in virus cell, so generally true cell structure is absent. So in virus, in crystallized particle, only two parts are present. So these are central core part is present. So it is a central core part. In central core part, genetic material is present. That means nucleic acids are present. So either DNA or RNA. DNA or RNA present in a central core. These central core nucleic acids are surrounded by protein coat. This protein coat is known as a capsid. In capsid, number of subunits are present. This is known as a capsomers. The study about a virus is known as a virology. The study about the viruses is known as a virology. So these viruses infect to bacteria, animals, fungus, plants, etc. etc. So virus infect the bacteria. These viruses is known as a bacteriophages. Bacteriophages. So phase is a nothing but a virion particle. Phase N is a nothing but to eat. That means infection causing viral particle is known as a phase. That means a complete infectious virus particle is known as a phase. So these phase virus virion infect to bacteria is called bacteriophages. Then these viruses, some viruses also can infect to cyanobacteria, blue green or algae. These type of viruses is known as a cyanophages. Cyanophages. So these viruses can infect to fungi also. This known as a mycophages. Then in the year of 1935, W.M. Stanley scientist, so first time crystallized the virus. So this virus is TME, tobacco mosaic virus. So tobacco mosaic virus was first crystallized by W.M. Stanley scientist from infected tobacco leaves. That means infected tobacco leaves of tobacco sap juice. Then along with the Northrop in 1946, they were awarded Nobel Prize in chemistry. So Stanley along with the Northrop scientists were awarded Nobel Prize in the year of 1946. Then history of virology. So developmental milestones in virology. So in the year of 1786, Edward Jenner scientist used vaccination for smallpox virus. So in the year of 1786, Edward Jenner scientist was vaccinated for smallpox virus. Then in the year of 1886, Adolf Meyer scientist demonstrated infection nature of this tobacco mosaic virus to tobacco leaves. How this tobacco mosaic virus infect? So what is the nature? So what are the symptoms, etc. These are demonstrated by Adolf Meyer scientist in 1886. So nature of infection of TME, tobacco mosaic virus to tobacco plants. Next one, in the year of 1892, Ivanovsky scientist proved these viruses are, that means which particles are infect to these tobacco plants, tobacco leaves, mosaic diseases. These particles are smaller than bacteria. That means virus are smaller than bacterial cells. So in the year of 1892, Ivanovsky scientist, viruses also first discovered by Ivanovsky scientist in the year of 1892. He also proved viruses are smaller than bacteria. Next one. In the year of 1898, Bacterial scientist defined the infection agent of tobacco mosaic virus as a Contagium vivum fluidum. Contagium vivum fluidum. So in the year of 1898, Bacterial scientist defined the infection agent to tobacco plants. So Contagium vivum fluidum. It is nothing but one living fluid material. So this living material, that means nothing but tobacco sap juice. This sap juice may contain some living chemical substances. These may responsible to cause the diseases to tobacco plants. So in the year of 1898, Bacterial scientist defined the infect agent to this tobacco plants as a Contagium vivum fluidum, nothing but living fluid. That means which fluid chemical substance caused to disease this tobacco plants. These might be living in a condition. In the year of 1984, Manchagner and Robert Gallo scientist discovered the HIV virus, AIDS virus. So in the year of 1984, Antigner and Gallo scientist discovered the HIV virus. So these viruses are smaller than the bacteria. Then size and shape of a bacteria. Size and shape. So viruses are ultra microscopic obligate intracellular parasites. These are smaller than bacterial cells. Diameter approximately 20 to 300 nanometers are present. So approximately 20 to 300 nanometers. So for example, bacteriophages. Bacteriophages, that means virus infect the bacteria, is known as a bacteriophages. So approximately 10 to 100 nanometers, 10 to 100. Then TMV, tobacco mosaic virus, so maximum 300 nanometers. Then based on the shape and the symmetry, so viruses are three types. So these are cuboidal viruses, then helical viruses, then complex structure or atypical viruses. So cuboidal condition. So cuboidal shape. Examples: herpes virus, then adenovirus. So cuboidal shape. Then helical symmetry, arrangement of protein codes are helical in a manner. So this viruses is known as a helical viruses. Example: TMV, tobacco mosaic virus, then influenza virus, helical shaped. Then complex shaped. These shows the some parts in a hexagonal condition, some in a helical manner. So both types of shapes may be mixed in condition. These viruses is known as a complex viruses, atypical viruses. Example: bacteriophages. Bacteriophages, then vaccinia virus. So head may be hexagonal condition, then tail fibers are arranged in a helical manner. Based on shape and symmetry, viruses are three types: cuboidal shape, helical shape, complex shape. Cuboidal shape herpes virus, adenovirus; helical shape tobacco mosaic virus, influenza virus; complex viruses, bacteriophages and vaccinia viruses. Then characteristic features of viruses. So viruses generally shows the both the living characters and non-living characters. So first one we have to see, so what are the living characters of viruses? So living characters, generally nucleic acids are present in a virus. So nucleic acid, so either DNA or RNA act as a nucleic acid, genetic material in a virus. So these nucleic acids are surrounded by protein coat. These called a capsid. So it is a one living character. So nucleic acid and proteins are present. Next, second one, due to this nucleic acid have the capability to mutations. Maybe changes takes place in genetic mutations. These mutations also rapid rate of mutations takes place in viruses. It can easily change its shape, that means capsid structure, proteins nature, that means number of the proteins are easily change and change the place also. Next one, So these viruses can active inside the host organism. That means replicate inside the host organism. Outside the host organism, it is an inactive condition. It is a non-living character. But inside the host, this may be active condition. Replication takes place inside the host. So active inside the host. So active inside the host, that means replication takes place inside the host. Next one, these viruses cause the diseases to plants, animals and human beings. So viruses can cause the diseases to all type of living organisms. All type of living organisms cause the diseases. It is also one living character. Next one, this shows the irritability. This shows the irritability. That means respond to its surroundings. That means environmental stimuli. So within the host only, not outside the host. So when this enter into host, inside the body of a host, so it responds to surrounding environment like a external stimuli. It is a physical, chemical or biological stimuli. So irritability like a consciousness, responsiveness. So virus shows the response to its environmental conditions. That means its surroundings inside the host only. Next one, it is a specific to host. It is a specific to host. So these are the living characters. Next one, non-living characters. So non-living characters are generally viruses are crystalline condition. So viruses are crystalline condition. So in that crystalline condition outside the host organism. So it is a one non-living character, not have the complete cell structure, acellular condition. Next one, metabolism also absent. Metabolism also, due to lack the metabolism, it cannot replicate outside the host organism. At the same time, enzymes also absent. Enzymes, due to lack of the enzymes, these cannot perform chemical reactions, metabolic reactions by itself, by own. So this requires the metabolism or enzymes mandatory for perform the all activities, replication, etc. That's why it is a obligate intracellular parasite condition. Next one, it do not shows the any functional autonomies. So any functional autonomies, these do not shows the any functional autonomies characters. So these are the non-living characters, both living and non-living characters. Next one, classification of viruses. Classification of viruses. Number of scientists were proposed the different types of classifications. But according to David Baltimore, so number of scientists were proposed the different types of viral classification systems. But according to David Baltimore 1971, 6 classes of viruses are there. That means David Baltimore scientist was classified the viruses into 6 classes based on genetic material. So DNA or RNA. So based on the nucleic acid. So what type of nucleic acid is present? So it is a DNA or RNA. It is a double stranded or a single stranded condition. So double stranded condition or a single stranded condition. If a single strand is present, it is a plus strand or a minus strand. So plus strand is known as a sense strand. Minus strand is known as a anti-sense strand. So generally in DNA, two strands are present. One strand polarity 3' into 5' and another strand polarity 5' and to 3' and the strand polarity with the 3' and to 5' and this strand is known as a template strand. This strand also known as a anti-sense strand. That means minus strand. So this also known as a non-codon strand. So template strand, non-codon strand, antisense strand, minus strand. Why? Because these strand not play important role in protein synthesis, mRNA transcription, etc. etc. These strand play this strand act as a template. That means copy the sequence from this strand to this strand only, complementary condition. Then with the polarity 5' and to 3' and this is known as a non-template strand. So plus strand, sense strand, then codon strand. So in this strand, the sequence is similar to RNA sequence, especially mRNA sequence, except in the place of thymine uracil is present. That's why this strand is known as a codon strand. So three adjacent nitrogen bases code for one amino acid. Based on this one, then code mRNA sequence, then polypeptide protein synthesis. If a single strand is present, this strand is a sense strand and anti-sense strand. Next one, RT, reverse transcription. Reverse transcription. Generally, transcription means DNA to RNA is known as a transcription. So the formation of RNA from the DNA, all types of RNAs, especially mRNA, is known as a transcription by the RNA polymerase enzyme. Then RNA to proteins, this is known as a translation. RNA to proteins, translation, transcription, then translation, central dogma theory of molecular biology. So transcription means DNA to RNA. Reverse transcription means RNA to DNA. This is known as a reverse transcription by the reverse transcriptase enzyme. If a single stranded condition, it is a plus strand or minus strand, sense strand or anti-sense strand. Then method of reverse transcription. That means method of production of RNA. Method of production. So DNA to RNA, then RNA to DNA, DNA to again RNA, then protein synthesis takes place. So based on these characters, so nucleic acids are DNA, RNA, these are single stranded, double stranded. If single stranded is present, it is a sense strand or anti-sense strand, plus strand or minus strand. Then reverse transcription method, then method of production of RNA. So based on these characters, Baltimore scientists classify the viruses into 6 classes. So first one coming to, if DNA acts as a genetic material, so it is a double stranded DNA. If double stranded DNA is present, these viruses are belongs to one class. So for example, adenovirus. Adenovirus. If a single strand is present, so these are the plus strand. That means sense strand of DNA. So these are parvovirus. Parvovirus. Then double stranded RNA. If RNA is a genetic material, so this RNA is a double stranded condition. Example, U viruses. Then single stranded RNA plus strand, that means sense strand. Example, Chaga virus. Then minus strand, anti-sense strand. Anti-sense strand. rhabdovirus. Rhabdovirus. Next one, reverse transcription in plus strand of and sense strand of RNA. If RNA is a single stranded condition, reverse transcription method plus strand. So RT method. So in this method, DNA intermediate in a life cycle. So reverse transcription in which viruses single stranded RNA is present, that means plus strand. So in this reverse transcription with DNA intermediate, that means RNA to DNA, so then DNA to again RNA and proteins. So here DNA intermediate. So DNA intermediate in a life cycle. So if single stranded RNA is present, it is a sense strand. Then reverse transcription, that means RNA to DNA, then DNA to RNA protein. So DNA is an intermediate in a life cycle. Next one, reverse transcription in double stranded DNA. If double stranded DNA act as a genetic material, here maybe reverse transcription takes place. So here with RNA intermediate condition. So these are the 6 classes of viruses, nearly 7 classes of viruses. So double stranded DNA, adenovirus, then single stranded DNA plus sense strand parvoviruses, then double stranded RNA reoviruses, then single stranded plus strand, so viruses minus antigen strand rhabdoviruses. Then if RNA is a single stranded condition plus strand sense strand, so in this reverse transcription with DNA intermediate life cycle. So these are one group of viruses. Then another group of viruses, if double stranded DNA is present, in reverse transcription with RNA intermediate condition. So based on these characteristic features, David Baltimore scientists classify the viruses into 7 classes. Next one, viral genome. So genetic material, nucleic acids. So generally the center core is a nucleic acids in a viral particle. It is either DNA or RNA. So generally genetic material is a linear condition, circular in a condition. So linear means end-to-end condition, end to end. So circular in a condition, there is no distinct end. So these are maybe joined in a condition. So linear or circular. So genetic material linear or circular. Generally in viruses, genetic material single unit of genetic material, entire maybe single chromosomic in a condition. But in some viruses, so for example wound tumor virus, then influenza virus or HIV viruses, some units are like a fragments like a present. So generally in virus, entire genetic material single unit of structure is present, but in some viruses, wound tumor, influenza virus, then HIV virus, like a fragments like a present, then 2 or 3 more units are present. Next one, in which viruses DNA act as a genetic material? These viruses is known as a deoxyviruses. So in which viruses DNA act as a genetic material? These viruses is known as a deoxyviruses. So RNA act as a genetic material. These viruses is known as a riboviruses. Generally in animals and fungal, and bacteria, animal, fungal and in bacterial viruses, DNA act as a genetic material. So DNA acts as a genetic material, animal viruses. But in some animal viruses, influenza virus, polio virus, HIV virus, RNA act as a genetic material. So animal, fungal and bacterial viruses, DNA act as a genetic material. But in some animal viruses, polio virus, influenza virus, in HIV virus, RNA act as a genetic material. So generally in plants, in plant viruses, RNA act as a genetic material. But in some plant viruses, wound tumor virus, cauliflower mosaic virus, then rice dwarf virus, DNA act as a genetic material. So generally in plant viruses, RNA act as a genetic material, but in some plant viruses, cauliflower mosaic virus, rice dwarf virus, wound tumor virus, DNA act as a genetic material. So these RNA is a double stranded condition or a single stranded condition. So double stranded condition, reoviruses. Double stranded RNA reoviruses. Then single stranded RNA TMV virus, tobacco mosaic virus. Next one, double stranded DNA. Double stranded DNA generally adenoviruses, then bacteriophages. Adenoviruses, then bacteriophages. Then single stranded DNA parvovirus, single stranded DNA. So these RNA, DNA, double stranded or single stranded condition. Double stranded RNA in reoviruses, single stranded RNA TMV, tobacco mosaic virus, then double stranded DNA adenovirus, bacteriophages, then single stranded DNA parvoviruses. Next one, ultrastructure of some viruses. So first one, TMV, tobacco mosaic virus. Ultrastructure of tobacco mosaic virus. So these tobacco mosaic virus cause the diseases to mainly tobacco plants, leaves of tobacco plants. This TMV was first discovered by Ivanovsky scientist. Ivanovsky scientist in 1982 from the tobacco plants, infected tobacco plants, tobacco juice. Then W.M. Stanley. W.M. Stanley scientist crystallized. W.M. Stanley. So in 1935, crystallized this tobacco mosaic virus. This tobacco mosaic virus caused the diseases to tobacco plants by some insects. So these are aphids, locusts. So aphids and locusts. So these TMV, tobacco mosaic virus, cause the diseases to tobacco plants by the insects, aphids and locusts, etc. Then disease symptoms are initially leaves or discolorization takes place. That means green to green mottling condition, then yellowish condition. That means lost the chlorophylls along with the veins. Discolorization of leaf along with the veins. That means this converted into yellow color or green mottling condition. Then rolling of leaves due to this one, these patches appears, so yellow color patches, so stunted the growth is reduced, so automatically yielding also will affect. In tobacco plants, leaves are the yielding, nothing but products. So due to infect to tobacco leaves, diseases appears, symptoms, patches like condition, yellowing patches, white spots like a condition. That means a discolorization of leaves appears, patches of yellowing or mottling green color. after the discolorization, then curling of leaves. That means will affect the photosynthesis. So reduce the assimilation of photosynthetic products. It will affect on growth of the entire plant. So growth is reduced, shows the stunted growth, reduced growth. Due to this one, decrease the number of leaves also. So yielding will affect. Then ultrastructure of tobacco mosaic virus, generally rod shaped condition, rod shaped helical virus. TMV is a rod shaped helical virus. So TMV is a rod shaped helical virus. Genetic material is a single stranded RNA is present. This single stranded RNA coiling around the central hole of a space. It is approximately 4 nanometers is present. So single stranded RNA act as a genetic material. This single stranded RNA coil around the center hollow space, approximately 4 nanometers. This contains the nucleotides, approximately 6500 nucleotides are present. The complete RNA required for replication of a tobacco mosaic virus, DNA. Around this nucleic acid RNA, protein coat is present. So it is a protein coat, capsid is called capsid. This capsid contains the number of proteinaceous subunits. This is called capsomers. Capsomers, approximately 2130 capsomers are present. 2130 capsomers. Then width of the tobacco mosaic virus, nearly 300 nanometers. Then length approximately 20 nanometers, 80 to 90, 20 nanometers. Then weight approximately 39 into 10 to the power of 6 daltons of weight. So tobacco mosaic virus, rod shaped helical virus. This was first discovered by Ivanovsky scientist in the year of 1892. So crystallized this virus by Stanley in the year of 1935 from infected tobacco leaves. So RNA, single stranded RNA act as a genetic material with 6500 nucleotides. This single stranded RNA coil around the central hollow space, nearly 4 nanometers. So complete RNA, single stranded RNA required for replication of this tobacco mosaic virus. Then this nucleic acid surrounded by protein coat. This is called the capsid. These capsid contain subunits, capsomers, nearly 2130 capsomers are present. Then diameter of TMA 300 nanometers, like the largest virus. Then length approximately 18 to 20 nanometers are present. Then molecular weight 39 into 10 to the power of 6 daltons molecular weight is present. Next one, bacteriophages. Bacteriophages. So ultrastructure of bacteriophages. So bacteriophages means virus infect the bacteria. This is known as a bacteriophages. So phasing. So phasing is nothing but to infect the bacteria. The virion particle infect to bacteria. This virus is known as a bacteriophages. So these bacteriophages first discovered by Twart scientist, Twart and D'Herelle. So Twart scientist first time discovered infection of a bacteria to virus. So infection virus to bacteria first discovered by Twart scientist in the year of 1915. It is also one milestone in virology. In the year of 1915, a Twart scientist discovered a viral infection to bacteria. Then the term bacteriophages was coined by D. Harley scientist in the year of 1917. In the year of 1917, D. Harley scientist discovered the or coined the term of bacteriophages. These bacteriophages generally shows the tadpole shaped. So bacteriophages shows the tadpole shape. So these shape contain both the hexagonal condition and helical. It is example for complex viruses. Then head in a hexagonal condition. These head the core with the DNA, double stranded DNA is present. These double stranded DNA 100 times larger than the length of bacteriophages. Then this nucleic acid double stranded DNA is surrounded by protein coat. This is called capsid. Protein coat is known as a capsid. Then tail is present. Tail sheath. So this tail hollow tubular like a condition. Tail and head are connected by collar. Tail and head are connected by the collar. Then end of the tail here one base plate is present. Base plate. Then 6 tail fibers, tail fins are attached to this base plate. Tail fibers. These tail fibers will be useful to recognize a particular place on bacterial cell wall, then attachment, pinning, landing on a bacterial cell. That means a host cell. So alpha structure of bacteriophages, it shows the complex structure. That means a tadpole shaped head and tail is present. This head and tail connected by collar. So head within the head arranged nucleic acid double stranded DNA. This double stranded DNA 100 times greater than total length of this bacteriophage. This double stranded DNA is surrounded by protein coat capsid. These capsid approximately contain 2000 subunits. Capsomers are present. So head and tail connected by collar. Then tail hollow tubular like a structure. Then end of the tail base plate is present. This base plate will be useful to inject genetic material into host organism like a bacteria. Next one, 6 tail pins, tail fibers are attached to this base plate or a tail. Next one, life cycle of a virus. So virus completes its life cycle by the 2 types of cycles. So first one is a lytic cycle, then second one is a lysogenic cycle. So virus shows the 2 types of life cycles, lytic cycle and lysogenic cycle. So lytic cycle is a virulent, then it is a non-virulent condition. So first one coming to lytic cycle. So lytic cycle is also known as a virulent. Why? Because at the end of the life cycle of this virus cause the lysis of bacterial cell, disintegration of a bacterial cell for a release. That's why this cycle is virulent cycle. This cycle cause to death of the bacterial cell at the end of a viral cycle. Why? Because for release this virion particle after complete mature from host required to lysis of bacterial cell by secreting the enzymes like a lysozyme enzyme. That's why this cycle is known as a lytic cycle, virulent cycle. This cycle will complete by 5 stages in a host. So for example, Escherichia coli. So first stage is a adsorption. Then second stage is a penetration. Then third stage is a synthesis. Then fourth one assemble and maturation. Then fifth stage release. So first one coming to adsorption. Adsorption is a nothing but this virion virus particle contact with the bacterial cell, then landing and pinning on a bacterial cell. That means bound with the bacterial cell is known as a adsorption. So in this first one, the virion particle. So the virion particle come and contact with the bacterial cell. So initially virion viral particle bacteriophages and come and contact with the bacterial cell. So bacterial genetic material. Then with the help of tail fibers recognize a specific place on a bacterial cell wall for landing. This is known as a landing. These tail fibers contain lipopolysaccharides. Lipopolysaccharides. By the help of lipopolysaccharides, these tail fibers recognize a specific place on bacterial cell wall. By recognizing specific place, land attach with adsorption with the bacterial cell wall. This is known as a landing. So after landing, the tail fibers are contrast. The tail fibers are contrast. Then the tail fibers are bending. The base plate enlarge. So tail fibers act as a anchoring. So after landing, these tail fibers are contrasting like a bending. Then enlarge the base plate. These tail fibers act as a anchoring. So this is known as a pinning. Chain landing and pinning. So first one is a adsorption. In this adsorption, these virion particle come and contact with the bacterial cell. By the help of tail fibers, these tail fibers contain lipopolysaccharides. So by the help of lipopolysaccharides, recognize a specific place on bacterial cell. Then bind on a bacterial cell. This is known as a landing. So after landing, this tail fibers bend, then base plate becomes enlarged. So tail fibers act as a anchoring. This is known as a pinning. So after absorption, then penetration takes place. So in penetration, the viral secretes the enzyme. This enzyme is known as a lysozyme enzyme. This enzyme will be useful to dissolve the bacterial cell. By dissolving the bacterial cell wall, inject the genetic material into host cell, bacterial cell. While this time the base plate becomes enlarged. Then the tail sheet contracts, so appears shorter and thicker in a condition. Then base plates inject the genetic material into bacterial cell, host cell. This is called penetration. So in penetration, when landing and pinning, after the landing and pinning, this virus secretes the lysozyme enzyme. By the help of this lysozyme enzyme, dissolve the bacterial cell wall. While this time the tail sheet become contrast, so appear shorter and thicker in a condition. Then base plates becomes enlarged. This base plate due to becoming enlarged easily inject the DNA, this viral DNA from bacteriophage to bacterial cell into bacterial cell. That means a host cell. This is called a penetration. So after penetration, synthesis. After this bacteriophage genetic material entered into bacterial cell, take over the whole metabolism of a bacterial cell. Why? Because virus lacks the metabolism and enzymes. Then suppresses the activities of a bacterial DNA. That means degrades bacterial chromosome, then bacterial proteins, then prevents, inhibits DNA replication of a bacteria. That means suppresses all bacterial activity, take over metabolism of a bacteria, then use the metabolism of bacteria, enzymes, etc. Then replication of viral proteins, nucleic acids, tail fibers, tail sheet, then base plates, etc. in usual manner, not completed whole of a structure in a single unit. In independent manner, all parts are synthesized separately. This is known as a synthesis. In synthesis, after inject the genetic material into host, these genetic material of bacteriophage take over the entire metabolism of a host. That means degradation of bacterial chromosome, DNA replication, then proteins of a bacteria. That means suppresses the all activities of a bacteria. By the take over metabolism of a bacteria, use that metabolism, then synthesis of a phase particles. All parts of phase virion independent manner is called synthesis. Next one, assemble and maturation. So in assemble, maturation. So generally in viral synthesis, synthesis or formation of viral parts, head separately, tail separate, collar separate, base plate separate, tail pins separately. In assemble and maturation, all parts come assembles to form a complete virion particle. Is known as a assemble and maturation. So after infection, within 20 minutes, they form the 300 virion particles. Assemble and maturation. So after maturation, again constant. So after the maturation, so secretes a lysozyme enzyme. This lysozyme enzyme dissolves the bacterial cell wall, cause the lysis of a host cell, then release the virion particles. So completes the lytic cycle of a virus. Next one, coming to lysogenic cycle. So lysogenic cycle is a non-virulent condition, not cause the immediate lysis of a host cell. So when this bacteriophage genetic material enters into host bacterial cell, this do not cause the degradation of bacterial chromosome, bacterial protein, then bacterial DNA replication. This viral genetic material incorporate with bacterial genetic material. When bacterial genetic material undergoes to replication, while the time of a cell division, it go replication along with the bacterial genetic material. That means not cause the immediate lysis of a host. That's why this cycle is known as a lysogenic cycle. So in this cycle also Adsorption, penetration is a common. Adsorption and a penetration. That means the bacteriophage come contact with the bacterial cell wall by the help of a tail fibers recognize the specific place. Then contraction of a cell's plate tail by using the ATP molecules. Then penetration and inject the genetic material into host cell. That events are common in lysogenic cycle also. So after inject the genetic material, so bacterial DNA, then phase DNA, bacterial DNA, then phase DNA. So bacteriophage inject the genetic material into host. That time it is a linear DNA. This linear DNA becomes the circular DNA. So phase DNA becomes a circular DNA. This circular DNA incorporate with the bacterial DNA. Do not cause the degradation of a bacterial DNA and metabolism of a bacteria. So this type of DNA is known as a prophase. Prophase means a bacterial DNA along with the phase DNA. That means a phase DNA incorporate with the bacterial DNA by the recombination. This DNA is known as a prophase. When bacterial cell undergoes to replication, when bacterial cell undergoes to replication, that means a cell division, that time this prophase also undergoes to replication. At every time of bacterial cell division, binary fission, the phase DNA also undergoes to replication, then synthesis of phase particles. For this one, it may produce the two receptor proteins. These receptor proteins will check the synthesis of a phase particle. So when bacteriophage inject the genetic material into host, in that condition it is a linear DNA. This linear DNA becomes a circular DNA. This do not cause the any degradation of bacterial chromosome duplication proteins. Then incorporate with the bacterial DNA by the recombination method. So this bacterial DNA along with the viral DNA is known as a prophase. At every time of bacterial cell division through the binary fission, this also undergoes to replication. This phase DNA may synthesis two receptor proteins. These receptor proteins checks the phase synthesis, that means virion particle synthesis. When these are post exposed to UV radiations and chemicals, UV radiations and chemicals cause the lysis of this host, then release the virion particles. These are ready to infect other healthy bacteria. So for example, Escherichia coli. So these are the life cycles of bacteriophages virus. So virion. So virion means a matured, complete infectious viral particle is known as a virion. So virion is nothing but a complete matured infectious virus particle is known as a virion. Next one, viroids. So viroids was discovered by Theo Diner scientist in 1971. Theo Diner. So viroids was first discovered by Theo Diner scientist in the year of 1971. So these viroids are without a protein coat, only nucleic acids are present. It is a single stranded RNA. So viroids is nothing but without protein code, only nucleic acids are present. It is a single standard RNA with a low molecular weight. Low molecular weight. So generally these viroids cause the diseases to plants only. Diseases to plant kingdom. Example, citrus, exocortis, then potato, spindle tuber disease, potato spindle. So viroids was first discovered by Theo Diner in the year of 1971. So viroids, nucleic acids without protein code, capsid is absent. So this genetic material is a single standard RNA with a low molecular weight. So these cause the diseases to plants. That means the plant kingdom. Best example, citrus exocortis, then potato spindle tuber disease. Next one, virusoids. So virusoids are similar with viroids, but small RNAs are present. Single standard RNA, but small RNAs are present. Small RNAs. These small RNA not a capability to replication in independent manner. It requires the support for replication. That's why the small RNA always link with the larger molecules of RNA, nothing but viroids, etc. This is known as virusoids. These are discovered by Randless scientists. So Randless scientists discovered the virusoids. These are similar to viroids. Contains the single standard RNA, but the small RNA is present. So these RNA not have the capability to independent replication. It requires the support for replication. That's why it always link with the larger molecules, virusoids. Next one, prions. So prions are discovered by Stanley and Prusiner, the year of 1982. So in the year of 1982, Prusiner and Stanley scientists discovered the prions. So prions are proteinaceous infectious agents to animals, not plants. That means nucleic acids are absent. Only protein code capsid is present. Only protein code capsid. Nucleic acids DNA or RNA is absent. Only protein code capsid is present. That's why it is a proteinaceous infective agent, especially cause the diseases to animals. Only protein code is present. Nucleic acids are absent. These cause the number of diseases to human beings and animals. So these diseases mainly affect on central nervous system of human beings and animals. Best example, Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. So Creutzfeldt-Jakob disease caused to animals to human beings. It will cause on symptoms on central nervous system. So next one, BAC, bovine spongiform encephalitis. Bovine spongiform encephalitis. This also known as a mad cow disease, caused to cows. Mad cow disease, bovine spongiform encephalitis disease. Then scurvy disease to sheep. So prions cause the number of diseases to animals, human beings. So best example CJD, Creutzfeldt Jakob disease mainly affect to central nervous system. Then BSc bovine spongiform encephalitis, this commonly called as a mad cow disease to cows. Then scabby diseases to sheep. So generally tulip flowers. Tulip flowers appears very attractive condition. Why? Because stapes are appears. So stapes and tulip flowers appears due to this tulip breaking virus. So tulip flowers stapes are appears. This is due to tulip breaking virus. That's why flowers are appearing very attractive in a condition, like a beautiful in a condition. It is nothing but one viral disease. This disease caused by tulip breaking virus. Stapes and tulip flowers. These belongs to OT viridae group. OT viridae group. Member of viruses useful as a insecticides, natural insecticides, like a bioinsecticides. Best example, baculovirus. Baculovirus, nucleopolyhedrovirus belongs to genus of baculovirus, baculoviridae. Next one, entomophax, then cytoplasmic polyhedrosis granulovirus. These are the best insecticides. So number of viruses will useful as a insecticides. Best example, baculovirus, nucleopolyhedrovirus. Best insecticide, narrow insecticidal program. Next one, entomophax, cytoplasmic polyhedrosis, granulovirus. Then viruses cause the number of diseases to plants, animals and human beings. So plant diseases. So basically mosaic disease. Mosaic, that means on leaves appears patches like a condition, leaves turn into a yellow color, etc. This disease is known as a mosaic disease. So plants are tobacco plants, then cauliflower plants, then sugarcane, cucumber, tomato, etc. Cause the mosaic diseases: tobacco, cauliflower, sugarcane, cucumber and tobacco. Then cause the bunching of banana, then potato, leaf roller, then leaf curling in a lady fingers, then Chumbro disease in a rice. So virus cause the number of diseases to plants. Mosaic disease to tobacco, cauliflower, sugarcane, cucumber, tomato. Bunching of banana, potato, leaf roll disease, leaf curling in a lady's finger, then leaf clearing, leaf veining, leaf clearing, then Chumbro disease in a rice. Number of diseases caused to animals. These are foot and mouth disease. Foot and mouth diseases to cattle. Foot and mouth diseases to cattle. Next one, rabies diseases to dogs. Virus cause the number of diseases to animals, foot and mouth diseases to cattle, rabies disease to dog, then encephalomyelitis horse. Like this, virus cause the number of diseases to human beings also. So these are common cold, hepatitis B, then SARS disease, severe acute respiratory syndrome, then AIDS, HIV virus, then mumps, rabies, measles, chicken pox, smallpox, chikungunya, then measles diseases. So virus cause the number of diseases to human beings also. In this common examples are common cold, hepatitis B, SARS, severe acute respiratory syndrome, then AIDS, HIV, then measles, rabies, mumps, chikungunya, chickenpox, smallpox, etc.